So this is a lecture on the connection between leaky gut, dysbiosis, and autoimmune diseases. And this is actually an expansion of a nano podcast that I gave last year. This is a lecture given for the Functional Medicine Symposium 2020. What is leaky gut? This lecture will discuss leaky gut or gastric permeability, and also the definition of dysbiosis or SIBO, small intestinal bacterial overgrowth, or as it should be called, small intestinal microbial overgrowth, and its connection to autoimmune diseases. We will also have a discussion of what types of testing is appropriate and some basics as far as early treatment options. So let's define leaky gut or gastric permeability. It's a medical condition where the intestinal lining in our villi, that's those little finger-like projections that go out into the lumen of the intestine, are more porous or permeable than they should be. The barrier between the outside world and our inside cells is disrupted. This barrier is made up of cells with tight junctions that are like the cement or the mortar between the cells. Think of it as a protective brick wall around a city. And if you disrupt this protective layer, or this wall, then an invading army can breach the wall of a fortified city, and this is what we call a leaky gut, or the invasion of things that shouldn't be allowed in, get in. When this leaking begins, it could be foreign material, proteins, large carbohydrate molecules, and the body's immune system kind of gets ramped up because these things are not supposed to be on the inside. They're considered invaders. And in war, we would call them an invading soldier or an invading force. In medicine, we call them antigens. And this normal innate immune response is what's mounted, causes inflammation, and then with prolonged inflammation, that's not very good for our cells. And then the adaptive immune system then kicks in, and then we produce antibodies. Now, we'll talk about how the antibody production can enact something called an autoimmune response. Because of something called molecular mimicry, at times our body's immune system, and we're talking about the adaptive immune system here with antibody generations from B cells, will attack our own tissue and organs, believing them to be the invading antigens. When this happens, it's called autoimmunity. Here we see sort of a cartoon of some cell walls. You have cell A on one side and cell B, or at least part of the cell wall, on the other side. And in between, you see where they are connected together. They're adjacent, but there's kind of like this glue or these tight junctions that are depicted by these little round blue things. And that's how it should be. They should be nice and stuck together. There should be nothing there that things can leak from one side to the other side through the extracellular space. And so this is a good depiction. You can keep this in mind when you have a discussion with your patients about leaky gut and what tight junctions are. I like to use analogies, and this is a picture of a European walled city. You can see the fortification, all the people are safe inside. Think of this as kind of like a cell, and it has a barrier on the outside that prohibits any kind of invaders from entering the city. Then you have a depiction, scanning electron microscope of an image on the upper right-hand corner of microbes. Those are bacteria there, and you can see some of the disruption that has occurred in between the cells that are depicted in sort of the yellow gold color. And since the disruption has occurred, bacteria are leaking through. Think of it as an invading army coming in. And things that we do in our lives, living not so healthy, eating foods that aren't good for us, like the standard American diet, imbibing in too much alcohol, not getting enough exercise, having low vitamin D, having low vitamin C and zinc, things like that can disrupt the integrity of our cell wall bonding, the tight junctions. And it's kind of like how the Trojans actually broke down their own protective wall to allow a gift from the Greeks to come in, the Trojan horse. And that was a big mistake because it breached their main line of defense and they paid the consequences. So what can cause leaky gut? Well, several things can cause. It's not just particularly any one single issue. It can be somebody's predisposition to have leaky gut in the first place. So one can be genetically predisposed. Some folks with more susceptible to leaky gut than others. Second, there may be some important triggers or aggravating factors, such as food sensitivity or food allergy. And third, it could be a disruption of gastric microbiome. So the normal bacterial balance in the gut is disrupted, and this can lead to a issue within the leaky gut system of tight junctions, and it's all based on the pathologic microbes. This can be most often established with acute gastroenteritis. Well, you can't really fix your genetics, at least not in today's day and age. That might be something of a future intervention, but we're sort of stuck with our genetics. So that leaves the other two modifiable reasons for leaky gut that we can address and fix. And we can do that by establishing the triggers. So if we know what triggers people have, we can identify those and remove those. Everyone's tipping point is different. Some folks can go years before a leaky gut manifests itself as a major medical problem, and some only require a few weeks to a few months, let's say. Putting out a big fire and keeping it from running away is a great way to stop a leaky gut from happening. So like we use the analogy of not throwing gasoline on a fire. When I talk about leaky gut to patients, I talk about their gut being on fire, and we have to douse it with water. We don't want to throw gasoline on it. So we need to identify triggers so we can eliminate that. That's the first thing you want to do. You don't want to dive into some complicated, expensive therapy if you haven't identified some of the triggers. And the other thing you want to tell your patients is that fixing the leaky gut at any point can take months to years to repair that and the collateral damage that can occur, i.e. autoimmunity. So in this cartoon, this illustrates a number of things. Things like stressors. At the top, you have antibiotics, which can be a stressor because it can disrupt the normal microbiome of our gut. You have food allergies or food antigens. You have pathological microbes, whether it's bacteria, viruses, or parasites. And then stress, physical stress, such as being in a car accident, or emotional stress, i.e. going through a divorce or losing your favorite pet. And then you have environmental toxins of all sorts. And it can be a combination of any one of these things or one of these things by itself, but usually it's in combination. People usually have some food allergy issues, and then they have stress on top of it and things get manifest. Now on the left middle part, the epithelial cells, you see the normal tight junctions. These cells are smack dab close to each other, and there's no room for leakage. So none of these microbes or food antigens or anything like that can leak through. To the right, you see sort of these cells are a little bit further spread apart. This is where the tight junctions are disrupted. And now you have bacteria and viruses that are leaking through and getting into the bloodstream. That's going from the outside to the inside. And then the transcellular, once the cell breaks down because there's so much GI inflammation, you have the disruption. That's that last cell on the right that's all red and it looks like jagged edges there. That's a cell that's kind of been destroyed, and boy, things will really leak through then. You have this waterfall, not just a drizzle, but a waterfall of microbes and toxins and things like that that are getting into the system. Once in the system, there's a disruption of our immune system. Its normal function is to keep the enemy out, and when we see a lot of these enemies coming in, it sparks a massive reaction. This reaction can be anything from a weakened immune system or a disrupted immune system, inflammation or cytokine storms, food sensitivity, and malabsorption and nutrient deficiencies. So what's the connection between leaky gut and autoimmune disorders? Well, I alluded to it earlier in this lecture, but I'll repeat it here. It's worth repeating. So there's somewhere between 100 and 120 different named autoimmune disorders, and I think we're adding a few every year as newer ones are being sort of recognized. Essentially, they attack different organs or tissues in different ways to manifest themselves by organ involvement. For example, an autoimmune attacks the brain tissue, the myelin sheath, let's say, and causes a breakdown. The breakdown is realized as plaques or white lesions on a MRI, and we give it the name multiple sclerosis. Another example is the autoimmune that attacks the skin, one of our largest organs, and this particular type of dermatitis is known as psoriasis. So that's another autoimmune disease. So you can call different organs or different tissues involved by the autoimmune hyperactivity and the confused immune system by whatever name you wish. It's not so important to me as to name it. It's more important to realize that this is what's happening, and it becomes involved with a dysfunctional innate and adaptive immune system. And then treatments are not to suppress or block as far as conventional medicine's kind of paradigm, but actually to offer therapies that will reverse or correct the problem of leaky gut and autoimmune disorders. Here we see a couple of cartoons. On the left, you have a normal body tissue and an invading microorganism, looks like a bacteria, that's in the gold. And then you have the antibody response. So those little Y-shaped things are antibodies that are attacking or sticking onto the antigens on the surface of that microbe. On the right, you have the autoimmune disorder. So you still have the microbe, you still have your Y-shaped antibodies, but you see where the antibodies are now attacking normal body tissue. So they're attacking to antigens that are similar to that's on invading microbe, and that's molecular mimicry. So the body's immune system kind of gets tricked into attacking itself. So how is SIBO connected to leaky gut? Well, SIBO, or small intestinal bacterial overgrowth, is often also referred to as dysbiosis. I kind of like that term a little bit better. Also, since it's more than just bacterial, we should be calling it small intestinal microbial overgrowth, since viruses and parasites and yeast and fungi can also be involved. These are then further broken down into subtypes. So you have two subtypes of SIBO, one which produces a certain gas called hydrogen gas, and that's called hydrogen predominant SIBO. The other has a different gas that's liberated from the gut flora, and that produces methane. So they call that methane predominant SIBO. And those are the two basic types. Methane, they say, is a little bit more difficult to treat. However, one can get a little bit distracted between differentiating the two. Let's just say, in the grand scheme of things, it really doesn't matter. And there's a lot of different testing, breath testing, and things like that are complicated and not very reliable as of yet, that people are pushing to get patients to do. I don't find them very useful in my practice. So when I diagnose SIBO, it doesn't matter whether it's methane or hydrogen predominant. I treat it as SIBO. Curing SIBO will eventually take one of the biggest offenders out of the picture of leaky gut. So leaky gut, as I mentioned, has a lot of different factors that are involved. If you can remove the dysbiosis and eliminate that, you can also help the healing process and restore the gut. Eliminating agitating or aggravating foods is another thing, and then cleaning up the body from toxins is one of the other things to focus on. real quickly, this is an image of the small and large intestines with a series of viruses, bacteria, parasites. This is just to illustrate what we have been talking about. So how long does it take to fix leaky gut? Well, that's a very good question, and it's not a very straightforward answer. It can take weeks and it can take years. A lot of times this is unrecognized because we're distracted with other things that seem to want to go to the top of the list, some autoimmune diseases that some fear may take a patient out quickly, and then you sort of trying to treat that, and it distracts you from underlying root causes. So you would want to consider a leaky gut as a sort of a root cause for some chronic illnesses or for some autoimmunity. It also depends on identifying the causative factors, whether it's bad food allergies or dysbiosis or some other heavy metal toxin. Sometimes dysbiosis can be resistant to therapy and can reoccur. Just because you licked it once doesn't mean that you may not have a recurrence a few months down the road. Somebody might have to go in for some antibiotics for a sinus infection, and that could throw the normal microbiome off again. One must follow very carefully the recommendations of a functional medicine physician to completely heal the gut. Conventional medicine, they kind of go off on a different paradigm than we do in functional medicine. This may involve strict dietary restrictions, such as elimination diets, or even worst case, an elemental diet. We can talk about that in a future lecture. Healing the gut may also involve recolonization of the gut with proper microbes and the use of agents, whether natural or antibiotic, to remove the bad microbes if that's necessary. Also, a healing or patching up of the tight junctions has to occur once the inflammation settles down. Finally, when the leaky gut stops leaking, one will then notice a return to health. This does not happen overnight, and again, it can take weeks to months. Healing can take months or even years for the gut health to return to a more normal picture. So a patient must have patience with the process. There's no magic speedy bullet, but you need to tell your patients to be patient and stick with the program, because eventually it'll work for them. If they give up on it too early, then they're going to set themselves back and they'll be miserable for a longer period of time. So the process of healing leaky gut can be quite lengthy, as I mentioned in the last slide. But the process of correcting an autoimmune disorder is even more tricky and can take even a longer period of time. You have to clear out those antibodies, and you have to make sure that you're not re-agitating the situation. Because the first step is to avoid rekindling the fire of the autoimmune disease. Leaky gut and other factors need to be fixed completely. Second, the way our immune system works with the T cells and B cells and with memory associated with those antigens, it can take quite a while. That is how our body actually continues to protect ourselves aggressively against foreign invaders, such as viruses, parasites, and microbes. So we have to kind of overcome our own natural protective mechanisms to see resolution of an autoimmune disease. So B cells that produce antibodies generate daughter cells that carry on the memory. Sometimes they're referred to as memory cells. These are sort of like the offspring of the original B cell. And those cell lines can propagate for many years. Throw in a new antigen every few years, and it kind of wells up the system. It's kind of like you've got your fire down to just little embers, and then you throw in an accelerant, something like gasoline, and all of a sudden you see that fire flare up again. So that's kind of what happens, and that's how tricky it is to actually treat this. You have to make sure you attach it in multi-factional ways and stick to it. If it's food allergies and environmental toxins, you got to address both of them concomitantly at the same time for this to all work. This is good if you are an invading nasty virus It is bad when it is an antigen, something like a gluten molecule. So our immune system should not be vilified here. It's actually there to protect us. It's to protect us from viruses and microbes, yes, but it should not be hyperactive when it comes to food antigens like the gluten or dairy molecules that can spark an autoimmune response. So again, it's multifactorial. We have to address each of these together in a big picture for us to heal. So in this cartoon, there's a depiction of B cells. You see B cells and some of the T helper cells and cytokines, which are chemicals that help with the adaptive immune system. So the innate immune system and assists the adaptive immune system cells like your T cells and B cells. And here towards the bottom of the slide, you see what we call memory B cells, which carry the memory of the initial antigen attack. And it's there again, that's how sort of the theory of how vaccines work. You give a low dose of an inactive form of a virus, and you hope your immune system will develop a memory to it and will be activated next time that virus appears. Same thing can happen with an allergy that develops against gluten or strawberries or peanuts. When there's a re-exposure, those memory cells come alive and will produce more antibodies, which will keep that sort of reaction alive. We'll talk a minute about testing. So I recommend an expanded food allergy testing, not only to check IgE or IgG, but actually IgE, IgG, IgG subtype 4, and a type of complement. That's one way to do it. The other more burdensome, complicated, and long-term way is doing an elimination diet, where you eliminate a lot of foods and then you slowly reintroduce, and you have to base it on your subjective feeling. That doesn't give hard data. It's not data. It's more of just subjective entries in your diary of what foods you might be sensitive to. The other thing is to check zonulin levels, either stool or serum zonulin, which is responsible for leaky gut. So the higher the zonulin level is, the worse your leaky gut is. Another thing you can check are LPS, lipopolysaccharide antibodies, and that's kind of the waste product, if you will, of gram-negative bacteria. So if those are sneaking through the tight junctions and getting on the inside of you and sparking an allergic reaction, that's a good indicator that the higher LPS antibody titers are, the leakier your gut is. Another thing you can measure is histamine and diamino oxide, which is the breakdown enzyme for histamine. That can be helpful when you're trying to determine if a histamine allergy is another factor. And then if you're not sure about a diagnosis of IBS, irritable bowel syndrome, versus IBD, that's a inflammatory bowel disease such as Crohn's disease or ulcerative colitis, and the person hasn't been scoped yet or biopsied, you can run some stool tests utilizing calprotectin and lactoferrin as non-invasive tools to determine if it's IBS or IBD, which can be part of this whole SIBO issue. A lot of people come in complaining of IBS symptoms, and that initiates the workup to begin with. The other thing we use is microbiome testing. So we check the stool for microbiology, or you can do a more elaborate CDSA, which is a comprehensive diagnostic stool analysis. Sometimes there's a lot of fluff in that, and it's not useful information, to be honest. And then ultimately, if somebody has not been scoped, and you want to make sure you're ruling out other nasty things, you can order a colonoscopy. So this is a report from a laboratory that we use in our center for the advanced intestinal barrier assessment, or what we like to call the leaky gut panel in our office. At the top you see zonulin. Now this is serum or plasma levels, not stool zonulin. And the measurement here is at 4.0, which is a little moderate elevation in the zonulin. So that speaks to us as a leaky gut marker. The more serum zonulin you have, the more leaky gut you have. The other things that are represented here is your histamine and your DAO. The DAO is an enzyme that breaks down histamine. So there appears to be an adequate level of diamine oxidase, but the histamine seems to be pretty high here. So it could be due to histamine-releasing foods or high histamine foods in the diet or some type of allergy. And you can also look at the DAO to histamine ratio, and that's actually pretty okay. So I would advise this patient who has maybe very mild leaky gut to avoid histamine-releasing foods or high histamine foods. And there are lists you can provide your patients with to give them guidance. Additionally, in the same report, we see measurements of LPS, IgA, IgG, and IgM. Now, in this particular patient, and this may not have been a good example to share with the group, everything seems to be pretty normal. The IgAs are a little bit on the higher end of the normal range, whereas the IgM's on the lower end. So when you look at this, you can't just take this at face value. You have to make sure that you're assessing somebody who, number one, puts out enough immunoglobulins. So you may want to check for total immunoglobulin levels in the serum before you do this. The other thing is, if you have a very high IgA, that means that the lipopolysaccharides that are the outer coating of gram-negatives are leaking through, and you're having an IgA response. Same thing holds true for IgG and IgM. Sometimes we see a low IgG level in the same picture with high IgA, and what's happening is the IgG is getting depleted. So either the patient's not producing enough or there's depletion because it's kind of very chronic and ongoing. Now, this is a report on IGG testing. So some laboratory tests for food allergies are IgE, and then there's some that use IgG. I tend to think that neither of them by themselves is very good or comprehensive. I like a larger panel that utilizes IgEs and Gs, in addition to some other things too. But here you have, this is categorized by types of foods. You have meat and dairy, poultry, fish, and seeds and nuts, fruits and vegetables down the bottom. And there's low IgG amount. So this is actually a person who doesn't have much issues with food allergies. In this panel of a different patient, you can see that we're measuring IgE here, and that's more of an anaphylactic type of reaction if they're very strong. We're also measuring a subtype of IgG. So IgG comes in pretty much 4 flavors, IgG 1, 2, 3, and 4. Interestingly, IgG4 has some blocking potential, and that's what this cartoon is representing here. So you have some moderate increase in the legumes and beans as far as food antigens go, and a low IgG4. So some of that IgG4 can actually bind to the IgE, and take it out of play. And you don't get that antibody attaching to certain cells and causing inflammation. So sometimes an elevated IgG4 is not a bad thing. In moderate and low situations, it might be kind of protective. But there are other tests that you will also want to rely on. Again, you can't take this at total face value. Here is yet another example of where we're measuring IgE and IgG4. And again, in the very bottom right on the right-hand side, you see the coupling or binding of the IgG molecules and the IgE. Again, taking those out. So if somebody were to have a moderate to elevated shellfish allergy but have moderate IgG4, it may be kind of protective. Again, trying to interpret this just by yourself is difficult. The information that's collected, which is massive amounts of information, is run through a computer algorithm so that it kind of analyzes all this data for you. Lastly, you can look at secretory IgA, and again, that occurs mostly in the mucous membranes. And you see here on this particular patient, there's some sensitivities of elevated IgA with relations to soybean high, and peanuts, navy beans, lima beans, a little bit of a moderate effect. Putting this all together is how I like to approach this. So I like a more comprehensive food allergy test that checks not only IgE, but IgG, IgG4, and complement, in this case, C3D, complement C3D. Now, again, looking at all this raw data can be a little bit confusing, and there is some assistance in the very right-hand side. You'll see in sort of a yellow column reasons for exclusion of why you want to exclude, let's say, almonds, is you have some blocking potential with the IgG4, but enough antigen IgE is present, reaction is present to that antigen to want to sort of consider that a sensitivity and possibly avoid it. If you look down at cauliflower, for example, cauliflower looks like it has quite a high IgE response, not much blocking protection from IgG4, and hence it's considered allergy and should be avoided. So there are sensitivities and allergies. And I tell my patients, hey, if you know for sure that you don't have anything listed to here, let's say blueberry. Let's take blueberries, for example. There doesn't seem to be any reaction with IgG, IgG4, complement, nothing. And you see here on the right-hand column, there's nothing to exclude you from eating this. But if you do know that after eating blueberries, your lips swell up and get pins and needles, or your ear canals start itching, then by all means, eliminate that food. It may be due to something else that's not being picked up by these tests for whatever reason. So that's just another thing to keep in mind. No single test is 100% perfect. The nice thing about Dunwoody Labs is that when they do these fairly comprehensive food allergy tests, they don't just leave you hanging with a 15-page report. The 2 pages within this report that is of most clinical relevance and for your patients is page 3 and 4. Page 4 is the more restrictive diet, and here we see the foods in the red column that should be eliminated. There are no foods listed in the moderate column, in the yellow-orange column in the middle. Those are foods that maybe want to take out early on and then reintroduce sooner than later. And then in the very left-hand green column are all the foods that are safe to eat. Once somebody is on the more restrictive diet for what I usually place people a minimum of 2 months, depending on the sensitivity and allergies, they might go 3 or 4 months. But usually I give them about a 2-month trial period on the more restrictive diet to get that under control before we segue into the less restrictive diet. So that's on page 3. Page 3 of the report would have this handout, which patients can print out and take them to the grocery store. And again, you saw the size of the number of foods in the red column is much more diminished here than in the previous slide. That means after a period of time, those other foods that made this list at one time can be reintroduced slowly, one at a time, and checked for any kind of subjective reaction. Once again, when you reintroduce foods with allergies, even though it has given the kind of green light to reintroduce it, if you do notice a sensitivity or a reaction, then you immediately pull that food back out, and it may be something you can never really reintroduce. The next thing I want to discuss is the stool analysis. So we've talked about a leaky gut panel. We've talked about analyzing for food antigens that might be triggers or things that kind of add fuel to the fire, if you will. But we want to see how bad the gut is as far as the microbiome, or how good it is. Let's say the glass is half full in this case. You want to make sure that you've got good microbes. So this is a stool analysis. This happens to be a sample report from DDI, and it's comprehensive stool analysis, so it's a little bit more complex, has a little bit more information than a standard microanalysis, so just the microbes. But here you have 3 columns again, the green, yellow, and red. Red, you really don't want anything in the red column. That could be things like Pseudomonas or Klebsiella, some really nasty bugs. It could also be things that are commensal in the yellow column that are 4+, and that moves actually to the red column. So right now we have nothing in the dysbiotic flora. We don't have any major issues with the gut as far as microbes go. There is one commensal microbe, alpha hemolytic strep, which is probably in all of our guts. And in this case, in this patient, there was enough of it to grow out what we call a 3+ colony. So you have NG, no growth, or 0, and then you have it all the way up 1+, 2+, 3+, 4+. You really want to be at 4+ on all the microbes identified in the green column. So if we look at the green column, you see there's no growth of a good Enterococcus species. There's only 2+ lactobacilli instead of 4+. And then Bacteroides, Bifidobacterium, and the good E. coli, there's only 3+ of those. So there is a little bit of a situation here where we're not getting the robust colony growth that you would see on an agar plate of these good microbes, and as a consequence, you have a little bit of an overgrowth of an opportunistic microbe that can throw this in a little bit of disorder. This report also looks at some digestion and absorption enzymes and markers, such as elastase, and so it looks at some muscle fiber and vegetable fiber in the stool and carbohydrates. And also looks at lactoferrin. Again, that's an inflammatory marker that can lead people to make a diagnosis of inflammatory bowel disease. And here we see at the very tail end of the report some illustration of short-chain fatty acids that are necessary for good stool analysis, and butyrates. The percent of butyrate is very important in good bowel condition. So this can be sometimes helpful in analyzing a CDSA. So now that I've finished discussing some of the simple testing that can be done, we'll move into how we fix leaky gut. And I'm going to talk about the basics here. Remember, this is functional medicine. It's very personalized. It's precision medicine, and no 2 patients are really alike, so you have to customize. But there's some basic things that you want to adhere to in this kind of paradigm of treating leaky gut that you should, consider on every patient, and then you sort of personalize it from there. So number 1, you want to definitely eliminate the offending foods identified in food allergy testing or sensitivity. You can do it by blood tests. You can do it by an elimination diet, but that becomes long, drawn out, and cumbersome. Okay, you want to use probiotics to recolonize the gut, especially if you have an abnormal microbiome microbe test and you have to replenish what's not there or what's weak. And use of higher doses of vitamin D3 as an immune modulator. Again, we're trying to help repair tissue and help protect our immune system, so we need an immune modulator. You can add in things like glutamine. This helps repair the tight junctions. The use of EPA or DHA, those are fatty acids found in fish oil, for example, as an anti-inflammatory agent. Because remember, when there's disruption and leaky gut, there's cytokine storms and inflammation that occurs. So you want to be in an anti-inflammatory position, not a pro-inflammatory position. The use of curcumin or other natural anti-inflammatory agents like quercetin or resveratrol to quell the fire can also be used. At times I'll use colostrum as a healing agent when indicated. And also potentially the use of low-dose naltrexone as an agent to suppress an autoimmune response and also to fix a dysfunctional immune system. So you have to have a good, healthy gut to have better LDN absorption, but you also can heal the gut because LDN has been shown in research to help heal a leaky gut and the mucosal lining directly. Of course, I always say You cannot throw gasoline on a fire. Some people use the analogy of a sink that's overflowing. Both the spigots are on and the water's just flowing into a sink that's clogged up. And you don't want to run in there with a mop and a bucket and try to sop up all the water before you turn the faucet off. You have to stop the flow before you mop up the floor. That's an obvious thing. Same thing here. You got to put out the fire, and you certainly don't want to throw gasoline on that fire. So turning off anything that insults or offends via food antigens and environmental factors are important. So start, again, if you're doing the food allergy panel and you get that 2-page clinical useful report out of the 15 pages, you want to start with the more restrictive diet plan first for the first 2 months. And then you kind of segue into the less restrictive diet, adding in one food at a time, like maybe every 3 to 5 days, making sure there's no noticeable reaction. And then, keeping highly antigenic foods, some things like dairy for some people, or soy or gluten. Sometimes you have to eliminate those completely. And avoiding high histamine foods. So if you have a test that comes back and you have either low diamine oxidase activity or you have high histamine, then a low histamine diet or histamine releasing foods or beverages need to be eliminated for that. And you have to find the right diet for each person. So I like to use the Institute for Functional Medicine's detox diet plan as a baseline. I don't typically make recommendations as far as diets go to my patients. I think diet's a 4-letter word, so I don't typically say, oh, go on the paleo or go on the Ornish or plant-based diet or this or that diet. I let them pick whichever one's best for them, because I certainly would not be able to make that correct decision most of the time. It's a very individual thing, and sometimes it's trial and error. But to give them some guidance, I give them the IFM. I print that 2-page diet plan out for them, hand it to them for at least a start. Again, another reminder that everyone is unique, and you have to stick with this precision personalized medicine to serve your patients best. Probiotics are a good thing to use. They're not for everybody, and if you have too leaky a gut and you use too high a dose, then you could create some more problems. So probiotics with or without digestive enzymes can be used. I like to use the BioCult product. Da Vinci Labs has a non-dairy chewable probiotic coupled with a digestive enzyme that's excellent. Healed people with reflux disease as well as dysbiosis with that one. The Claire Labs line, they tend to specialize in probiotic therapy, so their lines are pretty good. Life Extension has some phage therapy. So phages are some types of engineered good viruses that can take out the bad bacteria and leave the good alone. And then Megaspore Biotic is another spore-based type of way to replenish your microbiome without using active microbes that might be obliterated by stomach acid. Additionally, you can use standalone digestive enzymes to be used with each meal. And L-glutamine is another agent, an amino acid, natural, that can heal the tight junctions. I particularly like to use combination therapy with a product by Metagenics called Ultra GI Replenish, and it's a powder, and I start folks out with 1 scoop per day in a smoothie or shake, and then they can go up to using 1 scoop twice a day. In some cases, it can be used as an enhancer to a meal or a meal replacement. To reduce or eliminate pathological microbes, parasites, bacteria, the use of herbals such as grapefruit seed extract, uva ursi, berberine, S. boulardii, and others. Or in those really tough cases, we use Xifaxan and also sometimes Alinea. So in this slide, you see the documented L-glutamine and some of the combination therapies using colostrum, and a few others like aloe vera leaf and slippery elm bark extracts for the kind of cooling down of a gut on fire. Also, there are some other considerations. There's another medication that can be used that needs to be compounded. And if you see elevated levels of histamine and depressed DAO levels, you can use a pharmaceutical in a compounded form, ketotifen, which is an antihistamine, 1 milligram capsules. Make sure you specify gluten-free, dairy-free. And the instructions are to take 1 to 2 capsules TID, that's 30 minutes before each meal, for anywhere between 6 and 12 months, depending on the severity of the leaky gut. For pediatrics, you can make this into a suspension of 1 milligram per 5 mLs for children. Let's talk a little bit about retesting. I'm not always in a big hurry to retest. There are some practitioners that like to test people fairly frequently, fairly often. I'm not like that. Sometimes I don't even retest the stool microbiota. For instance, if we're having success with treatment, there's really no reason to recheck it. That's more of an academic pursuit if you want to see all those numbers and colonies come back up high. But if somebody's doing well, what's the point really? I do retest in about 6 months if they're not doing well. And sometimes patients will come to me and say, hey, can I get my stool tested at 3 months? I'm like, well, that's a little early. Let's give it a little time. So my philosophy is don't rush to retest too soon and don't rush to overtest. You can follow zonulin levels either in the stool or the blood, if that makes sense, to kind of gauge. It's a barometer for how bad the leaky gut is. It's a good thing if you start out with a high zonulin level and then you repeat it 6 months later and it drops a bit. Certainly that's a good thing to measure and give encouragement to your patients that things are going well. Checking up on stool micro about every 6 months, but not always. And it depends on what makes sense. If they're still having issues, yeah, then I'll check it. If they've had a bout of acute gastroenteritis or they've been on 3 rounds of antibiotics for a persistent sinus infection, then surely I want to check that stool micro again. And choose your food allergy testing panels wisely, or your vendors. There's several out there. Some are not very good, and then some are excellent. And I like to use the broader spectrum. Do I check that very often? Well, maybe one every couple of years, to be honest. So that concludes this sort of short lecture on the connection between dysbiosis, leaky gut, and autoimmune diseases, with an emphasis on leaky gut and the diagnostics and some of the basic treatments. And for those in attendance at this symposium, if you have any questions, you can email me at dr.salibi@Carolina Holistic Medicine. Please reference in the subject heading this conference and then say, you have a particular question here. Then I'll try to get back to you as soon as I can. If you have any questions about our clinical practice, I've listed here our website and also our toll-free number.